Firstly, a very special good morning to all present here. Myself Neha. I am a student of BSc Chemistry Honours Final Year from Nikiram Sharma Government College. And today video we are going to discuss about the glycolysis, Krebs cycle as well as respiration and fragmentation. Firstly, in glycolysis, glucose is converted into glucose 6-phosphate in the presence of enzyme hexokinase and ATP is converted into ATP. In second step, glucose 6-phosphate is converted into fructose 6-phosphate under the enzyme phosphoglutamate. In fructose 6-phosphate is converted into fructose 1,6-bisphosphate under the action of enzyme phosphofructokinase and ATP is converted into ADP. Fructose 1,6-bisphosphate is further converted into glyceraldehyde phosphate which is in isomerism with a dihap under the action of enzyme triose phosphate isomerase. Now 2 molecules of NAD positive is converted into 2 molecules of NADH and glyceraldehyde 3-phosphate is converted into 1,3-bis phosphoglycerate under the action of enzyme glyceraldehyde 3-phosphate dehydrogenase. Now 1,3-bis phosphoglycerate is converted into 3-phosphoglycerate and 2 molecules of ADP is converted into 2 molecules of ATP under the action of enzyme that is the phosphoglycerate kinase. 3-phosphoglycerate is converted into 2-phosphoglycerate under the action of enzyme that is the phosphoglyceromutase. 2-phosphoglycerate is converted into phosphoenol pyruvate under the action of enzyme enolase and release of 2 molecules of H2O. Now phosphoenol pyruvate is further converted into pyruvate under the action of enzyme pyruvate kinase and 2 molecules of ADP get converted into 2 molecules of ATP. Now we are going to discuss about the glycolysis. What basically glycolysis is? The term glycolysis has been originated from the Greek word that is glykos from sugar and lysis for splitting. The scheme of glycolysis was given by Gustav Amdam, Otto Merhoff and J. Perenaz. And is often referred to as the EMP pathway. In aerobic organisms, it is the only process in respiration. Glycolysis occurs in the cytoplasm of the cell and is present in all living organisms. In this process, glucose undergo partial oxidation to form 2 molecules of pyruvic acid. In plants, this glucose is derived from the sucrose, which is the end product of photosynthesis or from storage of carbohydrates. Sucrose is converted into glucose and fructose by the enzyme that is the invertase, and these 2 monosaccharides readily enter this glycolytic pathway. Glucose and fructose are phosphorylated to give rise to glucose 6-phosphate by the activity of the enzyme hexokinase. This phosphorylated form of glucose then isomerizes to produce fructose 6-phosphate. Subsequent steps of metabolism of glucose and fructose are seen. The various steps of glycolysis are depicted. In glycolysis, a chain of 10 reactions under the control of different enzymes take place to produce pyruvate from glucose. While studying the steps of glycolysis, please note the step at which utilization of synthesis of ATP or NADH occur. This is the diagram of the glycolysis process as I have explained earlier, and you can see it thoroughly. Here 2 molecules come into existence because this undergo isomerization, and this is the like a major molecule that take place in the reaction. ATP is utilized at 2 steps. First, in the conversion of glucose into glucose 6-phosphate, and second, in the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. The fructose 1,6-bisphosphate is split into dihydroxyacetone, phosphate and 3-phosphoglyceraldehyde. PGAL is converted into 1,3-bisphosphoglycerate, that is the BPGA. Two redox equivalents are removed in the form of two hydrogen atoms from PGAL and transferred to a molecule of NAD positive. PGAL is oxidized and with inorganic phosphate to get converted into BPGA. The conversion of BPGA to 3-phosphoglyceric acid is also an energy yielding process. This energy is trapped by the formation of ATP. Another ATP is synthesized during the conversion of phosphoenol pyruvate to pyruvic acid. Now, pyruvic acid is then the key product of the glycolysis. What is the metabolic fate of pyruvate? This depends on the cellular need. There are three major ways in which the different cells handle pyruvic acid produced by the glycolysis. These are lactic acid fermentation, alcoholic fermentation and aerobic respiration. Fermentation takes place under the anaerobic condition in many prokaryotes and unicellular eukaryotes for the complete oxidation of glucose to CO2 and H2O. However, organisms adopt kreb cycle, which is also called as aerobic respiration. This requires oxygen supply. Now, what basically is fermentation? In fermentation, say by yeast, the incomplete oxidation of glucose is achieved under anaerobic condition by sets of reaction where pyruvic acid is converted to CO2 and ethanol. The enzymes pyruvic acid decarboxylase and alcohol dehydrogenase catalyze these reactions. Other organisms like some bacteria produce lactic acid from pyruvic acid. The steps involved are shown in figure, and animal cells also, like muscular during exercise, when oxygen is inadequate for cellular respiration, pyruvic acid is reduced to lactic acid by lactate dehydrogenase. The reducing agent is NADH plus H positive, which is reoxidized to NAD positive in both the process. Here is the representation of fermentation process. That is, the glucose get converted into glyceraldehyde 3-phosphate and NAD is converted into NADH plus H positive. Now, glyceraldehyde 3-phosphate is get converted into 3-phosphoglyceric acid, which is then further converted into phosphoenol pyruvic acid. Phosphoenol pyruvic acid get converted into pyruvic acid. Now, pyruvic acid can also form lactic acid where NADH plus H positive get converted into NAD positive, whereas pyruvic acid can also get converted into ethanol, where in this step NADH plus H positive get converted into NAD positive. In both lactic acid and alcoholic fermentation, not much energy is released. Less than 7% of energy in glucose is released, and not all of it is trapped as high energy bonds of ATP. Also, the processes are hazardous. Either acid or alcohol is produced. What is the net ATP that is produced? What is the net ATP that is synthesized? Calculate how many ATPs are synthesized and deduct the number of ATP utilized during glycolysis when one molecule of glucose is fermented to alcohol or lactic acid. Yeast poisons themselves to death when the concentration of alcohol reaches about 13%. What then would be the maximum concentration of alcohol in beverages that are naturally fermented? How do you think alcoholic beverages of alcohol content greater than this concentration are obtained? What then is the process by which organisms can carry out complete oxidation of glucose and extract the energy stored to synthesize a large number of ATP molecules? Needed for the cellular metabolism. In eukaryotes, these steps take place within the mitochondria and thus requires oxygen. Anaerobic respiration is the process that leads to a complete oxidation of organic substances and the presence of oxygen and release CO2, water and a large amount of energy present in the substrate. This type of respiration is most common in higher organisms. Now we will discuss about the citric cycle. Here is the diagrammatic representation of the citric cycle. This is also known as C3 cycle. In first step, there is glucose plus fatty acids under the action of enzyme acetyl coenzyme A get converted into coenzyme A. Here, oxaloacetate get converted into citrate. This step is known as citrate synthesis and the enzyme is citrate synthase. Now, citrates get converted into isocitrate with release and consumption of water that under the action of enzyme that is the aconitase. Isocitrate get converted into alpha ketoglutarate where NAD positive is converted into NADH with release of one molecule of CO2 under the action of isocitrate dehydrogenase. Now, alpha ketoglutarate is further converted into succinyl coenzyme A. In this step also, NAD positive get converted into NADH. Here, coenzyme A also play an important role with the release of CO2 and the enzyme where it acts here is alpha ketoglutarate dehydrogenase. Succinyl coenzyme A get converted into succinate. Here GTP is converted into GTP which is further converted into ATP. Now succinate get converted into fumarate. Here FADH changes into FADH2. Here the enzyme which comes play an important role is the dehydrogenase enzyme. Now fumarate get converted into malate with the consumption of water molecule under the action of enzyme that is the malate get converted into oxaloacetate. Here NAD positive get converted into NADH under the action of enzyme that is the malate dehydrogenase. Now oxaloacetate further converted into citrate and in this way cyclic process occur. Thank you and have a nice day. I hope you really enjoyed this video. Thank you.